Hey everybody, welcome back. So now we're going to start talking about the plasma membrane and membrane transport. Plasma membrane is found in all cells. So every single cell type has a plasma membrane, which includes phospholipids that are linked together and proteins that allow for movement of materials as well as allow for different activities within the cell. Bacteria and eukarya, their phospholipids are ester-linked, and you can see what an ester-linked bond looks like. Whereas archaeobacteria, or the archaea, have phospholipids that are ether-linked. And so there's the difference between bacteria and eukarya as to the archaea. So the fluid mosaic model is a model that shows the ability of the plasma membrane to move fluidly, that's what I wanted to say. So it can move in that fluid environment. Our cells are surrounded by fluid. There's fluid inside and outside of the cells. The phospholipids are able to move within that fluid environment and stay attached through those ester or ether bonds. To maintain the plasma membrane rigidity, eukaryotic cells have structures called sterols, and you can see those sterols here, those little yellow structures. These are sterols. Sterols allow or keep the plasma membrane intact even though it is very fluid. The structure is not going to pull apart very effectively. In prokaryotic cells, and I won't say just prokaryotic, in bacterial cells, they don't have sterols, they have hopanoids. Hopanoids do the same thing as the sterols in your bacteria or in your eukaryotic cells. Archaea have no hopanoids, they don't have sterols, they have a more rigid plasma membrane, so they don't have the fluidity that our cells and bacterial cells have. But no matter which type of plasma membrane you have, there are going to be proteins that allow for different functions. So proteins are going to either span the entire plasma membrane, so that's what this is doing. These are integral proteins. They are integrated in the entire plasma membrane. Or we have some proteins that are found on either one side or the other side, and those are called peripheral proteins. But all proteins have different functions. So they may function in cell-to-cell communication. Our glycoproteins often are cell-to-cell communicators. They may function in sensing environmental conditions. They may function in transport of materials across the membrane. They may be enzymatic. There's a lot of different functions, okay? So the major ways that proteins work in transport is through either diffusion, osmosis, or in active transport mechanisms. You've learned about these in earlier classes, so I don't have a lot of information on each, but I will show you. Basic diffusion is the movement of molecules from higher to lower concentration. So you have these molecules up here in a high concentration, down here in a low concentration. So molecules move from higher to lower concentration down that concentration gradient until equilibrium is met. Simple diffusion, these molecules go directly through the plasma membrane. In simple diffusion, the molecules are small and nonpolar, so they can go directly through that plasma membrane. Whereas in facilitative diffusion, you're talking about molecules that are typically polar. And so when they move down their concentration gradient, they're going to move through a channel protein to allow the molecules to go through, even though the plasma membrane wouldn't allow them to go through normally. So remember, the plasma membrane functions in maintaining the environment inside and outside. So they don't allow certain molecules to go through. Plasma membrane is very selective. It's a selectively permeable membrane. Allows small nonpolar molecules directly through. Other molecules have to go through specialized proteins or come in different ways. We also have pumps. So a pump, a solute pump, is a pump that allows molecules to move against their concentration gradient using energy. And so here we have ATP. ATP is the energy currency that our cells use. And this is showing a sodium-potassium pump. So here we have a sodium-potassium pump in its relaxed form. In the relaxed form, 3 sodium ions can bind. And so I will mention that sodium is one of the most common extracellular ions, whereas potassium is one of the most common intracellular ions. And so sodium is inside the cell, and it binds to the sodium-potassium pump when the pump is in its inactive form. ATP binds and activates the pump changes in configuration, and the sodium moves out against its concentration gradient out of the cell. In this new configuration, there's a binding site for 2 potassium ions. So potassium moves in and binds, and then when the phosphate releases from the pump, so here you have the ATP is hydrolyzed to ADP plus a phosphate. The ADP leaves, the phosphate stays bound. When the phosphate releases, the pump goes back to its resting configuration, open to the inside, and the potassium is pushed inside the cell against its concentration gradient. So this is an active transport mechanism, and it's known as a pump. And this just happens to be the sodium-potassium pump, probably the most common pump that most people know of. Osmosis is the movement of water through a selectively permeable membrane from higher to lower concentrations until equilibrium is met. So it's basically diffusion of water. We're just moving water. There are 3 types of osmotic solutions that we typically talk about, and I'm showing you a figure from your textbook about osmosis in prokaryotic cells, but this could easily be with eukaryotic cells as well. The difference is in prokaryotic cells, there's almost always going to be that cell wall. In eukaryotic cells, depends. The cell wall may be there, or if it's an animal cell, there would be no cell wall. Okay. So here we have that cell wall. The cell wall is never going to change, but the cell plasma membrane, if water moves in and the same amount of water moves out, this means that the solution is isotonic. There's no net movement. There's always movement of water, but there's no net movement one way or the other. And so the cell itself is not going to change in size. If there's a hypertonic solution, so if the cell is placed into a solution that is hypertonic, then that means there is more water in the cell and less water in the solution surrounding the cell. That means there's more solute outside. And so water will move down its concentration gradient until we have equilibrium of water to solute ratio inside the cell and outside. And so as water leaves, in this case, the cell's plasma membrane shrivels. And the cell wall stays intact, but the plasma membrane pulls away from the cell wall, and this is known as plasmolysis. This also happens in plant cells when plants are placed in a hypertonic solution. In animal cells, the hypertonic solution will cause the cell to shrivel up or crenate. There's no cell wall, so the cell basically just shrivels and dies. So this is a mechanism of dehydration. And then here we have a hypotonic solution. So in a hypotonic solution, the cell itself has more solute and there's more water in the solution. And so water moves from the solution into the cell until equilibrium is met. And in this case, the cell is going to swell up. It gets bigger. But the cell wall around the cell protects the cell from bursting. So that's a benefit of having that cell wall. In plant cells, this is known as turgor pressure, and it helps plant cells to stand very erect. So you've noticed probably at some point in your life, you've seen plants that were wilted because they didn't have enough water, and you've seen plants that were standing very erect and looked really nice. Well, they had enough water, and so they had a lot more turgor pressure to hold them up. In animal cells, hypotonic solutions will actually cause the cells to burst because we don't have those cell walls to protect us. And then in animal cells, we also, or not, I won't say in animal cells, I'll say in eukaryotic cells, we also have transport via vesicles. And so this is specialized to eukaryotic cells. Bacterial cells do not use phagocytosis. So here what you're seeing is a mechanism known as phagocytosis. This is a form of endocytosis. These are all actually forms of endocytosis. Endocytosis is another word for, or is the mechanism of taking in substances by engulfing them with the plasma membrane. Okay, so you're pulling things into the cell and endo or inside the cell. Okay, phagocytosis is a form of endocytosis that I often call cellular eating, so that you take large molecules, and not all cells can do this. Not all eukaryotic cells can do this. But there are certain eukaryotic cells that do use this mechanism for eating. They take in bacteria, or they might take in other small structures into their cell, and then they can utilize that for nutrients. Or in the human body, some of our white blood cells do this, and that process of phagocytosis allows the cell to then break down bacteria and then present the bacteria for an immune response. So that would be another reason for phagocytosis. Pinocytosis is cellular drinking. So in general, all cells can undergo pinocytosis, all eukaryotic cells. The plasma membrane just makes a very small bleb off, or makes a small vesicle and pulls in fluids from the extracellular region with dissolved solutes. And then we have receptor-mediated endocytosis, which is a form of endocytosis in which molecules bind to receptors. So they have to be specific to those receptors. And once all the receptors are bound, then a vesicle that is coated pulls the materials into the plasma membrane or into the cell, forming what we call a coated vesicle that can then be stored or degraded, whatever we have to do. So this is actually how humans get rid of excess cholesterol from the blood. And then, ooh, this is a bad picture. I apologize. I didn't realize how bad this looked. So on my screen, when I was just looking at it, it didn't look this bad, I didn't think. But so this is exocytosis. So once you have a vesicle inside that has materials that need to be released, that vesicle can bind to the plasma membrane and become part of that plasma membrane, and then open up and release the materials to the extracellular fluid. So that is exocytosis. materials are exiting the cell. I'm going to stop here. This has been a 15-minute video. I think that's long enough. And we'll get into the cell wall in the next video. Bye.